First, I'd like to thank the conference organizer for inviting me to give this talk today. Here's my disclosure. So I think I'm going to start by giving you an introduction to the growth plate because that's the main topic of this whole symposium. So the growth plate is found near the ends of long bone where longitudinal bone growth happens in children. The growth plate is mainly composed of chondrocytes that are arranged in three histologically distinct zones in the growth plate called the resting zone, proliferative zone, and hypertrophic zone. Chondrocytes in the resting zone are stem cells or progenitor cells that can self-renew and also give rise to new clones of proliferative chondrocytes. These chondrocytes in the proliferative zones are then arranged in columns that are parallel to the long axis of the bone. And as they divide rapidly, they also gradually move down the column. And as they get toward the bottom of the column, they stop divide and start to undergo hypertrophy. This process of chondrocyte proliferation and hypertrophy lead to formation of new cartilage tissue, but simultaneously, the bottom of the hypertrophic zone is also being invaded by blood vessels and osteoblasts, which remodel the newly formed cartilage into bone, or the terminal hypertrophic chondrocyte may also directly transdifferentiate into osteoblasts. In either case, the net result is that new bone being progressively created at the bottom of the growth plate as chondrocyte proliferation and hypertrophy continues, and that leads to longitudinal bone growth. And the main focus on my talk is going to be how epigenetic mechanisms regulate this process. So I think epigenetics should require no introduction, but maybe I should clarify what I define as epigenetic modification for this talk. So by definition, there are heritable phenotypic changes without changing the DNA sequence. In the literature, it's usually used to describe changes in chromatin structure, histone modifications, or DNA methylation. Now, in some broader definition, it might also include microRNA, long non-coding RNA, or transcription factor binding. But for this talk, I would like to restrict myself to the narrower definition of epigenetics. And so I will not be covering some excellent work by Tetsuya Kobayashi's group and some others on microRNA, the role of microRNA in the growth plate. And I would also like to focus on how genes that directly regulate histone or DNA methylation affect human bone growth, and not talk about how some other interventions like malnutrition affect the growth plate through epigenetic mechanisms. So I will also not be covering some nice work by Marsha Phillips' group on how nutritional catch-up growth is mediated through histone deacetylase. All right, so histones are spools that DNA wraps around and pack into nucleosomes. And so histone modifications affect how tightly DNA binds and therefore affects the level of gene expression. For example, histone acetylation reduces the positive charge of histone, thereby weakening the interaction of histone with the negatively charged DNA. And so histone acetylation usually leads to a more open chromatin structure, increased accessibility to transcription factor, and therefore increase of gene expression. In contrast, histone methylation has a more complicated effect on gene expression. Depending on how many methyl group it's being added, it could be monomethylated, dimethylated, or trimethylated. And depending on which residue is being methylated, it could be associated with gene activation or gene repression. And then there's DNA methylation by DNA methyltransferases. DNMT1 is involved in gene imprinting, which I will not be covering in this talk. But DNMT3 is involved in de novo DNA methylation, which is a mechanism that was used for gene repression. So I think one good example of epigenetic regulator regulating the growth plate is histone deacetylase HDAC4. HDAC4 haploinsufficiency causes a human growth genetic disorder called brachydactyly mental retardation syndrome. The role of HDAC4 in the growth plate has been elucidated by some mouse models. The HDAC4 knockout show growth retardation with ectopic bone formation and early hypertrophy, while the transgenic mouse overexpressing HDAC4 show the opposite phenotype with a lack of bone mineralization and delayed hypertrophy. So these mouse phenotypes suggest that HDAC4 suppresses chondrocyte hypertrophy in the growth plate. Now, HDAC4 is histone deacetylase, and so deacetylate histone and likely lead to repression of gene expression. It turns out that HDAC4 represses some genes, including like RUNX2, MEF2Cs, that are transcription factors important for chondrocyte hypertrophy. And so you can imagine the lack of HDAC4 in the knockout lead to upregulation of these transcription factors, therefore leading to early hypertrophy, while too much HDAC4 in the transgenic lead to suppression of these genes, therefore causing delay or impaired hypertrophy. And more recently, it's been found that HDAC4 is involved in mediating the effect of parathyroid hormone-related peptide, or PTHrP, in suppressing chondrocyte hypertrophy. So, as you may know, PTHrP is produced primarily in the resting zone of the growth plate, and it diffuses across the growth plate, forming a gradient. So this gradient, also because it suppresses hypertrophy, it also determines where hypertrophic zone starts at the growth plate. And it turns out that PTHrP, this effect was mediated by dephosphorylating and therefore activating HDAC4 in the growth plate, which also help explain why HDAC4 haploinsufficiency and PTHrP mutation in humans share a very similar skeletal phenotype. Now, I think HDAC4 provides us with a very good example of how epigenetic regulator can play an important role in the growth plate. But more recently, there are a lot of attention being given to the connection between epigenetics and human overgrowth syndrome. And so I'll spend a large part of my talk focusing on this. So there was a study with 710 patients that found mutations in epigenetic regulators being the major cause of overgrowth syndrome with intellectual disability. In particular, there are three phenotypically very similar overgrowth syndrome that they all involve mutations affecting either histone methylation or DNA methylation. Soto syndrome is caused by NSD1 mutation affecting H3K26 methylation. Weaver syndrome is caused by EZH2 mutation affecting H3K27 methylation, while Tettenbaum-Raman syndrome is caused by mutation of DNMT3A, which is a DNA methyltransferase. And of these three syndrome, our group focused on the Weaver syndrome because of a patient that we saw a few years ago at the NIH. And I'm going to tell you more about that. So EZH2 is part of the polycomb repressive complex, also known as PRC2. The PRC2 catalyzes the H3K27 trimethylation, which is a repressive histone map. And in 2010, there was a genome-wide association study, GWAS, that identified a large number of genomic loci, including the EZH2 locus that is associated with normal human height variation in the general population, suggesting that common variants of EZH2 might affect how tall a person becomes in the general population. And then 2 years later, in 2012, EZH2 mutation was found in patients with Weaver syndrome. Around that same time, we saw a patient at the NIH who has the one on the right-hand side here. He has overgrowth syndrome and he has an advanced bone age. We performed exome sequencing on this patient and the parents, and we found that he has a de novo heterozygous missense mutation of EZH2, confirming that he has Weaver syndrome. Now, these patients are tall, but they have normal levels of growth hormone, suggesting that the role of the function of EZH2 is probably mediated directly through the growth plate, which is why we got interested and we want to study it further. So we generated a knockout mouse model. We use a catalyst-specific EZH1 and 2 knockout, considering that there's functional redundancy between EZH1 and EZH2 in making the PRC2 complex. So this EZH1 and 2 double knockout show growth retardation with decreased body weight and T-billing starting at around 3 days of age. When we look at the growth plate, these knockout mice have decreased chondrocyte proliferation, the column, decreased length of the column, and there's decreased chondrocyte proliferation as shown by BRDU staining, which label the cells in the S phase of the cell cycle. We also saw that the hypertrophic chondrocytes are not as big, suggesting they were unable to fully undergo hypertrophy. So we try to understand the mechanism of these growth plate phenotype. And so we perform laser capture microdissection first with the proliferative chondrocytes, and we study the transcriptome. As you might expect, because EZH2 catalyzes a repressive chromatin map, the loss of EZH2 leads to upregulation of a large number of genes and pathways in the growth plate, including one pathway that affects the cell cycle. And so we hypothesize that EZH2 probably play a role in controlling the cell cycle in the growth plate. In particular, we want to study genomic locus that contains 2 cyclin-dependent kinase inhibitors, CDKN2 and B, that binds to and inhibits CDK4 and 6 to block cell cycle progression. We found that H3K27 trimethylation, this repressive chromatin map, is normally enriched at the CDKN2 and B promoter. And the loss of this map in the knockout chondrocyte is associated with an increase of gene expression. We then confirm that this effect is mediated through EZH2 because when we use an inhibitor to block EZH2 activity, we found an upregulation of CDKN2 and B expression, which is associated with a decrease of chondrocyte proliferation. And importantly, this effect of decrease of chondrocyte proliferation can be partially rescued if we simultaneously knock down CDKN2 and B expression using an siRNA, therefore confirming this causal relationship. We then try to do a similar study in the hypertrophic zone, again using laser capture microdissection. And again, we found a large number of genes and pathways being upregulated in the knockout. And this time, we found that one of the pathways that was being upregulated was downregulated was IGF signaling. So, without going into too much detail, I just want to mention IGF-1 signaling is an important pathway for chondrocyte hypertrophy. For example, IGF-1 knockout mice have significantly smaller hypertrophic cell size. And I'm just going to go directly to the conclusion here. We found that EZH2 positively regulate IGF signaling by repressing the expression of something called IGF binding proteins, including IGF BP3 and 5. So the downregulation of IGF-BPs by EZH2 free up the ligands like IGF-1 or 2 to interact with the IGF-1 receptor, therefore stimulating IGF signaling. And so taken together, EZH2, we found that EZH2 plays an important role in both the proliferative zone and the hypertrophic zone, in part by allowing the cell cycle progression and also positively modulating IGF signaling. And when we're studying this EZH1 and 2 mice, around that same time, another group in Boston were actually studying another mouse model which is the EED knockout. Now, EED is another member of this PRC2 complex, and the EED knockout mouse has a very similar skeletal phenotype as ours, except they were focusing on other pathways. They were focusing on the Wnt signaling and TGF-beta signaling. And so putting ours and theirs together, it looks like PRC2 complex plays an important role in the growth plate by mediating or controlling many different signaling pathways that are important in the growth plate. So now the EED mouse or our EZH mice didn't look anything like the Weaver syndrome. So what is going on? We thought about that. One hint is that Weaver mutation or Weaver syndrome is caused by heterozygous missense mutation of EZH2, but rarely do you find that these mutations are early truncating mutation or frame shift mutation, suggesting that Weaver syndrome cannot be caused by a simple loss of function of EZH2. And that's supported also by our mouse models because the loss of EZH2 activity doesn't give you overgrowth. It leads to decreased growth in the growth plate. And so we start to wonder, at least at that point, it wasn't so clear whether this mutation a gain of function or loss of function. And so we start wondering maybe we can study it to see if it's gain of function mutation or loss of function mutation. And it turns out that studying EZH2 activity is not as straightforward as we hope, mainly because EZH2 catalyzes multiple steps in this enzymatic reaction. In some other system, for example, B-cell lymphoma that also involves EZH mutation, they found that the people who study B-cell lymphoma found that EZH2 mutation lead to the mutation abolish the first step in this reaction, the monomethylation step, but it actually have enhanced activity in the second step, the dimethylation step. And so if you study the mutation on its own, it will look like a loss of function because it can't perform this first step of this reaction. But if you study it in a heterozygous state, it would actually turn it into a gain of function mutation because the wild type was able to perform the first step, sort of a coordination between the wild type and the mutation. And so when we study the Weaver syndrome, we should have this in mind. So we decided first study the Weaver mutation in some EZH2 knockout chondrocytes that doesn't contain endogenous EZH2 activity. We found that the Weaver mutation has an activity that is somewhere between the mock transfection and the wild type EZH2, therefore suggesting a partial loss of function. We then performed the same experiment with some wild type chondrocyte, this time with endogenous EZH2 activity. And basically they have same level of EZH2 activity, which argue against some kind of coordination between the wild type and the Weaver mutant that would turn it into a gain of function. So it's partial loss of function. We then try to confirm that by generating a knock-in mouse carrying one of the Weaver mutation in our patient. The homozygous mutant died perinatally, but the heterozygous mutant was healthy and it actually has a site overgrowth. So it kind of recapitulated the human overgrowth phenotype. And we were able to use this mouse to study histone methylation activity. We found that in both the homozygous and the heterozygous mutant embryo, that there's decreased EZH2 activity based on decrease of H3K27 dimethylation and trimethylation staining. therefore confirming that the Weaver mutation is indeed a partial loss of function. Okay, so if it's really a partial loss of function, why weren't there more early truncating mutation of EZH2 in the Weaver? So what is the function? What is the clinical consequence of a heterozygous non-mutation of EZH2? That's the other way to put it. We thought about that. So one possibility is that the heterozygous EZH2 non-mutation is clinically silent. But that's unlikely because when we look in the general population, the loss of function EZH2 mutation is extremely rare, suggesting there's a strong evolutionary selection against it. Second possibility is that the mutation is lethal. It certainly is in the mouse, but when we look in the Decipher database, we actually found quite a few patients with heterozygous deletion that contains the EZH2 gene, suggesting the heterozygous loss of EZH2 is actually compatible with life. And so we think the most likely explanation is that the heterozygous null mutation of EZH2 lead to a clinical phenotype that is distinct from overgrowth. So if you look at this chart, we think the heterozygous Weaver mutation, which is kind of a hypomorphic mutation, lead to decrease of EZH2 activity, and that lead to the repression of some genes, presumably growth promoting genes. Therefore, it gives you overgrowth. The heterozygous null mutation gives you 50% EZH2 activity, and that leads to the repression of even more genes. Now that gives you more problem. For example, maybe gives you organ dysfunction, decreasing fitness. So it gives you something, but it just doesn't show up as overgrowth, and so you don't see this mutation in Weaver syndrome patient. And now we think one remaining mystery of this epigenetic and overgrowth syndrome is that why all three, or even potentially more, overgrowth syndrome all involve the mutation in epigenetic regulators, but they seem to be modifying different epigenetic marks. Could there be some unifying mechanism that could tie the three syndromes together? So I think I'll spend the last couple of minutes just mentioning a few recent studies that seem to have found a connection. There's a 2019 Nature paper that found a connection between H3K26 dimethylation and DNMT3A. It turns out DNMT3A has a PWP domain that recognizes and binds to H3K36 dimethylation. In embryonic stem cell, H3K36 dimethylation and DNMT3A were found to colocalize in some intergenic region. And when you knock down NSD1 in these cells, which is like Soto syndrome, you have a loss of H3K36 dimethylation, which is expected because that's what NSD1 catalyzed. But then you also see a loss of DNMT3A recruitment to the same genomic region, suggesting that in both Soto syndrome and Tetramer-Raman syndrome, there could be a decrease of intergenic recruitment of DNMT3A, and which then therefore leads to hypomethylation of intergenic DNA. Now, it becomes harder to explain how that leads to overgrowth, but at least it looks like there could be a common pathophysiology. And the same is true for Weaver and Soto, because there's another study that found NSD1 can bind to PRC2 complex, which then affects the genome distribution of H3K27 di and trimethylation. And so it looks like at least it's conceivable that there's a common pathophysiology between the three overgrowth syndrome that all involve the NSD1 driving H3K36 dimethylation, which in turn affect the genome distribution of other histone chromatin mark that is affecting the other two syndrome. And I think I'm gonna just end here and show my acknowledgement slide. And I would like to thank you all for your attention, and I look forward to the question and answer session. Thank you.